I am very, very honored and delighted to be joined by Tom Ben-Arye today, who is a senior scientist at the Good Food Institute Israel. He is a graduate of the Technion, that's where he did his Ph.D., and he actually pursued a career in academia so that he could help advance technologies in cultivated meat. Tom's findings were recently published in Nature Food and also helped form the basis of the first Israeli cultivated meat company, Aleph Farms, that many of you are aware of. So I think this is, it's an understatement to call Tom a brilliant scientist, but a little known fact about Tom is that he's also the most prolific hummus lover in the world, and as a really delightful cat that I hope will join us today named Benjamin. He likes to kind of flit across the screen. Yeah, it has really just been the greatest honor to be Tom's teammate, and I'm really excited to learn from him today. So I will stop blabbering now. Tom, without further ado, please take it away, and thank you again so much for being here with us today.
Hi everyone, hi Amy, thanks for inviting me over. I will just note before I start that a few days ago there was another paper that was published in Nature Food by Professor Mark Post, Professor Shulamit Levenberg, my PI at my PhD, and Professor David Kaplan. So I think this is a really exciting thing. It's a review article that explains about several of the issues with cultivated meat that needs to be addressed. So I haven't had the chance to read it yet, but I think it's really exciting and worth noting before we start this lecture about another Nature Food publication. So I will be speaking with you today about my PhD on bovine skeletal muscle tissue engineering, a brief introduction, why do we even need cultivated meat? So intensive factory farming and poor animal welfare are a cause for foodborne illnesses, such as the avian and swine influenza. And in addition, they also contribute to the spreading of E. coli, salmonella, and campylobacter that can be found in meat. And we think that if we can develop meat in a clean environment, we might be able to solve these kind of issues that are associated with meat currently. In addition, these are some of the problems that we see today. However, in the near future, we will see some more devastating problems. The high use of antibiotics in this industry, 70% of the antibiotics are being used by animal agriculture. And this is a very big concern for us because there are estimates that say that by 2050, the antibiotic-resistant bacteria will kill more people than of cancer today. Will also have a heavy toll on our economy of about $2.9 trillion per year for the OECD countries. So quite a big issue. So if we can find ways to produce meat in a cleaner way that doesn't require that much antibiotics, that's something that could be very useful for humanity. And it's not something all that futuristic. If you heard of the last resort antibiotic colistin, there are now bacteria which are resistant to that because pig farmers use colistin on their pigs, and this antibiotic resistance started to spread throughout Asia and South America. Also, a few years ago, there was the case where a woman came to the hospital in Nevada, and the physicians realized that she had bacteria in her body that were resistant to all known antibiotics combined. And after a few days, she passed away because there was no way to treat her. And we want to make sure that these kind of incidences won't become much more frequent. In addition, if we talk about the environmental impact, I won't go too much into the details, but from the aspect of greenhouse gas emission, this industry pollutes more compared to all of the transportation combined. If we talk about resource use, you need 15,000 liters of water to produce 1 kilogram of beef. Just to make sense of this number, for domestic use, domestic use uses 5% of our water consumption. whereas animal agriculture uses 55% of our fresh waters. In addition to that, 40% of the arable lands are being used for meat production, and by 2050, meat demand will double. So essentially, we just need to find more efficient ways to produce our food. It's just not very sustainable. Lastly, this industry slaughters about 100 billion animals per year in conditions that most people don't even want to hear about, which is another reason why we have an incentive maybe to change this industry and make it without the use of animals. So we are going to discuss today cultivated meat. Just a brief reminder or some kind of, for those of you who have no idea what this is, the basic principle is taking a biopsy from an animal. From this biopsy, we isolate the cells of interest. One of the advantages of cells is that they can proliferate. So from this small batch of cells, we can let them proliferate until we get tons and tons of cells, which we can then differentiate. Differentiate means maturing the cells, turning them into mature tissues. We can turn them into mature tissues, and then we can process them into food products. All of this sounds very simple, but let's see a little bit of how this can be done. So in order to develop something like that, we need to understand what meat is. Meat is a skeletal muscle tissue. It contains mostly muscle fibers, but also connective tissue, the extracellular matrix, fat, and a little bit of blood. In our research, we focus mainly on the muscle fibers and the connective tissue, basically saying that for cultivated meat, we need to include ECM producing cells to create this important portion of the meat. In addition to that, cells have lots of cell-cell interactions. They communicate with one another. They have synergistic effects. And because of that, co-cultures are important in order to develop a native tissue, just like inside the animal. You cannot just have one cell and expect it to behave as if there are many types of cells in there. Lastly, we need to grow cells in an environment that mimic their native conditions. If you think about a cell in your body, it's in a 3D environment, in a gel-like environment. For that, we use scaffolding. You can see here a plastic-based scaffold, the PLLA-PGA. It's basically a porous material which has some kind of mechanical forces, mechanical rigidity to hold the tissue in the correct side. It's also porous, so the cells can go inside. And we combine it with some kind of a protein gel, the fibrin gel, that gives the cells the biochemical environment that is similar to what they have in the body. So the cells can go inside and develop native muscle tissues. Previous work before of our work you either use the bovine monocultures of a single cell type or co-cultures of human or mouse cells in co-culture models. And if we talk about the scaffolding, other works either didn't use a scaffold at all, use the plastic-based scaffold, a sugar-based scaffold, or an animal-based scaffold, which is doesn't really solve the problem if we use materials from animals. In our work, we use the bovine co-culture, bovine satellite cells, supporting cells. Supporting cells are cells that produce the extracellular matrix, the ECM, and endothelial cells that form vascular networks. We culture these cells on an edible scaffold, on texture soy protein, TSP or TVP, and that was our approach for this research. So a little bit about TVP or TSP. It's a meat substitute, which is the main reason why we chose it. We want to start with something that is already somewhat similar to a meat that already mimics it. If you can compare it to the plastic-based scaffold, no one is going to ever use it. Plastic, hopefully, no one is going to use it. So we started off with a meat substitute. A TSP is also protein-based, rich in protein. It's very inexpensive. It's a byproduct of the oil industry. This material is also porous, as you can see here. which is really one of the reasons why we chose it, because it really mimics the structure of the other scaffolds that we use. And this is something important for the cells. And it also has versatile product geometry, as you can see here, which help us think that maybe we can, because of the way that extrusion works, the way that we produce the TSP, we can think that perhaps we can design it to be suitable for a bioreactor and developing different geometries that are suited for the final process. Our hypothesis in this research was that TSP can be used as a cost-effective scaffold for bovine skeletal muscle tissue engineering, and that co-culture with support cells and endothelial cells will enrich the ECM content and mimic natural muscle tissue more accurately. The objective of this research was to characterize the TSP as a tissue engineering scaffold, to optimize the culture conditions to create bovine muscle tissues with enriched ECM, and investigate the effect of cell co-culture on the ECM proteome. So I will start off with presenting the first objective of characterizing the TSP as a scaffolding material. We first took the scaffold and put it inside the scanning electron microscope to see the surface of the TSP in the interstructure. A scanning electron microscope allows you to visualize things in a nanometer scale. The first thing that we see, you will see it further on better. The first thing we see is that the TSP has pores which have some kind of directionality, which is something that is important for myotube alignment and to get the actual texture of meat. If we look at the inner structure of the TSP, you can see that on the micron scale, everything seems flat. And on the nanoscale, we see that the TSP is composed of these 30 nanometer ball-shaped, probably protein clusters. And if we look at the surface of the TSP, you can see that on the micron scale, it is a little bit more coarse. But if we look deeper inside, we see that it has some kind of an amorphous glue-like structure. I don't know, this is something just a basic characterization. If we look at something more interesting, for example, the TSP pore size, we see that the majority of pores are in the range that are suitable for cell culture, about 50 to 400 microns, where we can grow the cells. And we also have a few dispersed larger pores that are larger than 400 microns that we think that can theoretically be used for media perfusion, maybe in someone's next PhD. You can try and do something like that. And that's something that you have several pores here and there, and for future perfusion, that's something that is important to have. Basically, because these large pores cells cannot really close them, and because of that, they are important as well. We next went to characterize the TSP structure on the 3D structure of the TSP. We took two types of TSP called Archon and TVP. Doesn't really matter. The main goal of using these two types of TSP was that we didn't want to check one type of TSP that is in a very specific conditions, but we wanted to show that there are things which are broader, that you can see that happens in different types as well. So we took one TSP1 that has 70% protein, and TSP2 that has 50% protein. We noticed that we knew from a colleague in the lab, Idan, showed that protein can absorb iodine and can be used in that way to visualize them in micro-CT. We tried that and we tested that, and we saw that it works also with the TSP scaffolds. So basically, we stain them with iodine, and the micro-CT allows you to see heavy atoms such as iodine and have good contrast with them. So you can see here a visualization of the TSP. You can see here, I hope you can see here the pores of the TSP. This is the entire scaffold. And if you can see well, you can see that the pores have some kind of alignment, which is something that is sought after when you are trying to generate skeletal muscle tissues. So this is the visualization that we get with the micro CT, and then we quantified it both for TSP1, TSP2. We also measured the PLLA, PLGA scaffolds that are commonly used in the lab for skeletal muscle tissue development. If we look at the pore interconnectivity, this is something important because we don't want to have pockets inside of the scaffold where the cells can penetrate. So one of the parameters we measured was the pore interconnectivity. We saw that almost the entire pore volume is well connected, over 98%. If we look at the porosity, the void content of the scaffold, it is about 50%, meaning at the end product, we will have about 50% of the scaffold, 50% of the volume still the scaffold itself, which is rather important for the nutritional value and the texture, something to think about. In addition to that, we measured the surface area and found it is about 100 centimeters per ml, and those are the micro-CT characterizations. Next, we want to check how the cell seeding efficiency works on the TSP to make sure that when we see the cells remain on the scaffold, don't fall off. Because if the surface is too closed, then the cells cannot penetrate into the scaffold. So we measure it in different conditions with 500,000 cells, with 1.5 million cells, in co-cultures, in tri-cultures. And in all of the conditions, we saw that even when we do it in a manual labor with a lab assistant, which is not very GMP conditions, we still always have above 80% seeding efficiency, which means that the scaffold can be well seeded through the experiments. In addition, because we want to use the scaffold not just for small scaffolds in the lab, we want to also make sure that we can see the thick tissues, because at the end we want to scale up this process. So we took a TSP chunk of about 1 centimeter. We seeded it with cells, and which were red. And a few hours later, we cut it and saw the inner and outer surface. And as you can see here, we didn't quantify it, but you can see that the cells are both in the inner and on the outer surface, in a very similar manner, meaning cells can penetrate into thick TSP samples. In addition, I think this is the last portion of this part. We wanted to check if cells can attach to the TSP and if they can spread on the TSP. So we put a very small amount of cells, just 50,000 cells, and we let them go over 3 weeks. And you can see here how the cells spread and attach and spread over the entire TSP. These results made us very happy. Of course, each line that you see here is a cell, and you can see also the pores of the scaffold inside of the TSP scaffold. If we cut the scaffold in order to see the cell at different section, so we used cryosectioning in order to cut the scaffold, and used H&E staining to visualize the cells and the scaffold itself. You can see here the red is the scaffold, and those small lines are the cells. You can see the cells both in the top, bottom, middle, and bottom of the scaffolds, also inside the pores, not just on the scaffold itself, meaning we generate a dense tissue that basically removing the void area of the scaffold, at least to some manner. And basically, to summarize this part, we showed that the TSP had over 80% seeding efficiency, and that when we perform the seeding, the cells penetrate into the thick TSP samples. We see that cells can adhere and proliferate on the TSP, that the pore size is suitable for cell culture. We did, however, saw a partial degradation of the scaffold during the cell culture, and the TSP also have a high autofluorescence, meaning it's rather difficult to perform the experiment there. So in some cases, we just perform the experiment elsewhere before doing it with the TSP, and this is something important for you to know. Also, additional information, the surface area of 100 centimeters per ml a 50% porosity, and the pores are directional and are interconnected. We also replaced the sterilization that we commonly use in the lab from ethanol sterilization to gamma sterilization. This is important because we are going for a food, something that is meant for food production, and ethanol sterilization is just not something that you can do there. And lastly, TSP2 compared to TSP1 had higher degradation and was more uniform in the z-axis. I didn't want to go into that. It doesn't really matter, because our goal is not to compare TSP1 to TSP2, but rather to understand the process itself. Next, we will go into the myogenesis, the way that we differentiate cells into myotubes. Basically, we start with the bovine satellite cells. Those are mature stem cells of the skeletal muscle tissue that can fuse and turn into myotubes. So I want to really thank Professor Marsha Hathaway from Minnesota University that provided us cells and protocols. She was just wonderful in that part. I would note that you can see here cells that we cultured in 2D using those protocols. And however, these protocols also have matrigel inside it, which is not food grade, and also some carcinogenic materials. So we remove them obviously because they are not suitable for cultivated meat research. And this entire process has 2 parts. I will go into it a little bit later. And basically, we tried this protocol also on the scaffold, and we got horrible results. You can see here the scaffold in blue and the cells in red. They really didn't grow well. We didn't even try to measure myotube formation because it's irrelevant. We thought maybe this is the end of the project because we didn't know what to do. But then before we threw everything away, we tested it also on the PLLA scaffold, which are suitable. We know that we can generate tissues on them. And we can see that also in the PLLA PLJ, we see that cells didn't grow well. If we try to look at the myotubes, all of them seem round. So we were happy that at least basically what we understood is we just need to optimize the differentiation protocol for 3D environments. So we use some information from the literature, and some information from Marsha Hathaway's protocols. Basically, the understanding that bovine satellite cells, they proliferate in higher concentration of EGF and IGF-1. And if we want to induce myogenesis, we can induce it with starvation, with IGF-1, and the PFGF inhibits the myogenesis progression. And we also understand that the myogenesis have 2 steps for this protocol. The first protocol is the expansion phase with the proliferation media, where we want to have the tissue as dense as possible so the cells can fuse afterwards, and also the differentiation phase in which the cells fuse and turn into the myotubes. So and this happens in starvation conditions, so we want to have this stage really short as possible, because it's not good for the cells at all. So we designed this full factorial experiment, design of experiment type of experiment. Basically, we had 3 factors that we changed during this experiment. The proliferation media, either the regular one that we used before, or the new type of media that we use called LLM1. We compared these 2. We also compared with or without IGF in the differentiation media, and with or without EGF in the differentiation media. So we have these 3 factors that we change, and we quantify the cells using 3 quantifications: the percent of myotube coverage, how much they cover the entire scaffold, the average myotube length, and the myotube shape complexity. Low complexity means a rounded myotube, whereas a high shape complexity is more elongated. So here you can see the results, and we were really happy to see that. Basically, you can see the proliferation media on the left and the differentiation media on the top. You can see that the LLM1 and LLM2, their combination showed really nice myotubes that were formed. And if we quantify it using the three quantifications that we showed before, the myotube shape complexity, the average myotube area, and the myotube coverage, you can see here the control. This is what we started with that looked really horrible, and this is the best conditions that we had. If we compare them, you can see that the average, the percent of coverage of myotubes had almost fourfold change, that the myotube average area increased about twofold change, and the myotube shape complexity increased in threefold change. So that's something that made us really happy. And when we analyzed it using the factorial statistical analysis, we saw these results. Factorial analysis allows you to get much more data, both on the factors themselves and on the interaction. Something really interesting to try to implement into your research. Gives you much more data with much less samples. Basically, if I will make everything shorter, we can see that the proliferation media, the LLM1 versus the control, improved all of the parameters that were measured in a very good p-values. Similarly, the IGF had in the differentiation media had very large effects also on all of the measurements. And also these two factors, the proliferation media and the IGF in the differentiation media, had synergistic effects, meaning not only each of them contributed, but their combination contributed even more. So both the proliferation media that we used and the IGF in the differentiation media, both of them are really important. We also saw some effects of the EGF on the average myotube length and especially on the myotube shape complexity. So we continued on using them. However, I can tell you that because our goal is to lower the cost, perhaps adding this EGF is not that necessary in the differentiation media. So we tried to take this proliferation media, the LLM1, and we tried it on the TSP scaffold. All of the experiment before, I don't remember if I mentioned, but all of this experiment was done on the PLLA PLGA scaffold just because it was much more convenient to do all of this analysis. And I will continue on and say that we tested now this new proliferation media on the TSP scaffold. You can see here in the new proliferation media and the old one. And you can see that in the new one, the cells are much more spread on the entire scaffold. They look much healthier compared to the ones in the control that we had before. And we see that the cell coverage on the scaffold increased from 18% to 72%. The dye that we have here is a dye that is, we internalize it into the cells at the beginning of the experiment. And when it is found inside of the membrane of the cells, so if cells proliferate, the amount in each cells go down. So if you see here a cell which has a very high intensity, means that it also didn't proliferate, whereas here if you can see a lower intensity of color, it means that the cells are in a better, that they have also proliferated. Something just to know, not that very important, but something to see here as well. And next we differentiated the cells. We put the cells inside also differentiation media. We see that we were able to form myotubes, multinucleated, elongated myotubes inside the TSP scaffold. It made us very glad. However, something to note is that the coverage of the myotube, the amount of myotubes that were formed, was very low. About, consider the numbers, about 2 to 5%. This is really not enough. And our thought was, that if we perform co-cultures, this would obviously become much better. So to summarize, in the media optimization part, we remove the matrigel and the carcinogenic materials. We saw that LLM1 and LLM2 improved myotube size, complexity, and coverage on 3D scaffolds, the PLGA scaffolds. We showed that LLM1 improved the bovine satellite cells coverage and morphology on the TSP scaffold, reaching over 70% coverage, and that LLM1 and LLM2 facilitated myogenesis on TSP scaffolds. Next, we went on to investigate the co-cultures of the cells. I will note that in this part was mainly done by Dr. Yulia Shandalov-Levy and Dr. Shachar Ben Shaul, which really helped with the co-culture experiments. So the cells that we used in these experiments are for muscle progenitor, use the bovine satellite cells. For the supporting cells, the cells that produce the ECM, we use the bovine aortic smooth muscle cells. And for the endothelial cells, we use bovine skeletal muscle microvascular endothelial cells. The choice of the supporting cells was both because they produce the ECM and from the fact that smooth muscle cells are cells that naturally support endothelial cells. So that was the logic behind using them. I think that using other type of supporting cells, perhaps fibroblasts, specifically embryonic fibroblasts or skeletal muscle fibroblasts, might have interesting results as well. Perhaps even a combination of those supporting cells could be really exciting. So this is for you to do. So you can see here the co-culture results on TSP1 and TSP2. This is the monoculture of the bovine satellite cells, the co-culture with the endothelial cells, the co-culture with the supporting cells, and the triculture, the combination of the three cell types. We quantified here both, you can see here the cells in red, the nuclei of the cells in blue, and myogenin is a marker, it's a transcription factor found inside the nuclei of mature skeletal muscle fibers. So basically we use the myogenin in order to estimate how mature the cells are. So here is the quantification. You can see that the basically we see the highest myogenin nuclei count in the co-culture with the smooth muscle cells and in the triculture. Where if you look here, these colors will be constant. The co-culture with the smooth muscle cells, the red will also look afterwards as one of the best results out there. Well, maybe I'm telling secrets for later on, but we also checked the myogenin also in a Western blot, showing that also here, both the co-culture with the smooth muscle cells and the triculture were much higher, significantly higher compared to the monoculture of the bovine satellite cells, also were significantly higher. In addition, we looked at the ECM that the cells deposited. We checked both the empty scaffold, the bovine satellite cells monoculture, the two co-cultures, and the triculture. Usually, we perform the cryosection and trichrome staining. Usually, with the trichrome staining, you can see this light blue color, which is usually accounts for collagen deposition. However, we had very high noise from the scaffold itself. Even in the empty scaffold, we saw some signal. So we didn't associate it to collagen, but we said that it's some kind of a protein deposition of some sort. And again, here we can see really nice results with the co-culture of the smooth muscle cell compared to the other ones, which just makes sense because when you add the ECM producing cells, you will see more protein deposition. We can see that this group was higher, significantly higher both compared to the empty scaffold and the monoculture of the bovine satellite cells. And because this was not very accurate, we also went on and tested a Western blot of collagen type 1. Again, you can see that both the co-culture with the smooth muscle cells and the triculture, the bovine satellite cells, bovine smooth muscle cells, and the endothelial cells, both the co-culture and the triculture were higher compared to the monoculture of the bovine satellite cells. Also, were significantly higher in both cases. And we also measured the scaffold weight as compared to the initial weight of the scaffold. When we started the experiment, we weighed the scaffolds, and we weighed them also at the end of the experiment. You can clearly see that the bovine satellite cells, the monoculture, lost a heavy amount of the weight, and it was really nice to see. And the scaffold themselves, we also saw that they were rather brittle. It seems that the bovine satellite cell monoculture really tore apart the scaffold, and it was really hard, really difficult to quantify them as well. However, it was really nice to see that in the co-culture, especially with the co-culture with the smooth muscle cells, we saw that the scaffold were much less brittle, and that in some cases they even increased their weight compared to the starting measurement. So that was really wonderful for us to see how these co-culture really affect the results. We also perform a test of mechanical properties. This was done mainly by my colleague, Dr. Shira Landau from the Levenberg lab as well. We compared the empty scaffold, the bovine satellite cells, the co-culture of the bovine satellite cells with the smooth muscle cells. We also compare it to the native bovine muscle. So we see that in the young model, we see that the basically shows how well, the opposite word of well, how oh, sorry. We see that when we look at the young model, we see how much the cells can, how much the scaffold can stretch. Basically, how strong how I lost my words. I will go to the UTS, the ultimate tensile strength, and then I will come back to it. So basically, the ultimate tensile strength, this is the more important factor that we care about. Basically showing us how unbrittle the scaffold is. Basically, we see that the scaffold maintain their structure when we have the co-culture with the smooth muscle cells, and without it, the scaffold really becomes brittle and break without even trying that hard. And the young model also shows us how much the scaffold can deform, how hard it is to deform the scaffold. So without the co-cultures and with the monoculture, the scaffold can really deform. And I would say that these results are not that comparable to the native bovine muscle, because it doesn't really check the elasticity. When they deform, they don't come back, which is kind of a problem with these cases. But when we look at basically the important part of the young model is that we are roughly on the same magnitude as compared to the native bovine muscle. Compared to other scaffold, it's right in the right conditions. So in summary, when we talk about multicellular seeding on the TSP scaffold, co-cultures with the smooth muscle cells and triculture showed the elevated myoglobin expression, elevated collagen type 1 deposition, and higher scaffold weight, and improved the construct integrity. And lastly, our final experiments was trying to assess the proteome of the tissues that we formed. I will note two things before I will start speaking about this, that we chose to do these experiments on the PLLA PLGA scaffold, because when you try to measure proteins, it's very unwise to start with a protein scaffold, because basically you will get lots of background noise, because if you take 10 micrograms of protein from the tissue, most of it will be the scaffold itself. So we said we prefer to use a scaffold that is not made from protein. Theoretically, you can also choose to do transcriptomes on the TSP scaffold because transcriptome measures RNA, and RNA measurements are much more sensitive to begin with, and also you don't get a noise from the scaffold from the RNA in the scaffold itself. However, we wanted to test the ECM, and if you want to test the ECM is something that is accumulated. And if you measure the transcriptome, perhaps the ECM is deposited in the first week, in the first week and a half. And if you will measure the transcriptome after 2 weeks, perhaps there is not much protein deposited at that stage. So if you want to measure a protein that is accumulated, it just makes sense to use a proteomic approach. So for this, we still use the PLLA PLGA scaffolds. So we did this test. For those of you who don't know, this is a volcano plot. The higher the point is, the more significant the measurement is. Each point here stands for one type of protein, and points that are on the right mean this is a log scale. Points that are on the right are higher, have a elevated expression in the co-culture, and points on the left have elevated decreased expression in the co-culture. So a point here is more in the co-culture, point here is more in the monoculture, and just remember that this is a log scale. 0 means that they are equal, and 10 means that it's 2 to the power of 10 higher in the co-culture compared to the monoculture. So if we look at the proteins that were elevated in the co-culture, we see lots of proteins related to the ECM, a collagen cluster, a laminin cluster, lots of additional proteins related to the ECM. You can see here a list of them. I won't go through all of it, but related to the ECM, we see TGF beta, collagens, laminin, fibronectin precursor, collagen fiber assembly, and so on and so forth. If we look at the myogenesis related proteins, we see TGF beta again, guide the F, L, and B, which guide proper skeletal muscle development, tropomyosin, myosin light and heavy chains. Really things that we loved seeing all of this. We really appreciated this. And the important thing in proteomics is not to look on a single protein, because there is lots of errors, lots of false negatives and false positives. Basically, you want to look at trends. You don't want to look at one protein. This is the big advantage of proteomics, that it allows you to look at trends, whether something shifts, whether a certain group of protein shifts to the right or to the left. So if you look at the groups of proteins related to the ECM, microfibril binding, heparin binding, ECM organization, we really see a shift to the right, meaning that they are mostly found in the co-culture, that they are higher elevated, that those type of proteins are more expressed in the co-culture compared to the monoculture. If we look specifically on collagen-related proteins, we see also a shift to the right. Some proteins here which are higher elevated, that are higher expressed in the co-culture. Also things related to the sarcomella, basal membrane, and basal lamina, we see some kind of shift to the right. Believe me, it's kind of significant, but we see here something also, some kind of a little bit of a trend, and a little bit also in proteins related to actin filament and strands fibers, but mostly in the ECM. So in summary, we characterize the TSP for cultivated meat scaffolding. We show that LLM1 and LLM2 enhance 3D myogenesis. Co-culture and triculture improve DCM production and myogenesis, and the proteome analysis showed strong evidence for complex ECM development due to co-culture with the bovine smooth muscle cells. So before I finish, I want to thank everyone in the lab, especially Professor Shulamit Levenberg, Dr. Yulia Shandalov-Levy, Dr. Shafar Ben Shaul, Dr. Shira Landau, Dr. Yedidia Zaguri, and Iris, who are now continuing this project as well. To the best lab engineer in the world, Galia Ben David, Idan Radensky, that helped with the iodine experiments, the micro-CT, to our lab manager, Dr. Inbal Michael, and to Janet, who helped with the, Janet Zavin, who helped with the trichome statements. So, and before I will finish, I will also say special thanks to Aleph Farms. This project began Aleph Farm and afterwards was funded by Aleph Farm. Special thanks to Dr. Neta Labon that really helped with designing some of the experiments and giving us wise advice. The smaller proteomics center and the BSF bioimaging center. And special thanks to Professor Mark Shahadaway that really helped us at the beginning, to Professor Mark Post, who really started this entire field and was really there and really initiated everything. And the special thanks to the Good Food Institute. I added it before I joined the Good Food Institute, so I think it is okay to still have it here because they really help a lot making all of this field be accelerated to the way that it is. So I want to thank everyone for listening, and if there are questions, I will be happy to answer them.
Thank you so much, Tom. That was so good. I'm going to stop sharing your screen now so that people can see your face. That was such a phenomenal seminar, informative and also like beautiful. I think like a lot of what you showed was really, really striking. We have a lot of questions that have come through here in the chat, along with a lot of people who've just raved about what an educational seminar that was. So a lot of people thanking you for sharing your knowledge with us. We have our first question here coming from Neves. Since the textured soy protein is not functionalized with RGD peptides, how do you think the cells adhere to it? Would the hypothetical addition of RGD improve cell attachment, in your opinion?
So the scaffold itself is made from protein. So I guess that there are many types of places where the cell can just attach to there are so many proteins there that come from the basically, usually if you talk about synthetic scaffold that are made from some kind of polymer, so the cells don't really have attachment points. But here, when you have so many proteins developing the scaffold, composing the scaffold, they can just attach to the protein of the scaffold itself. It could be that functionalizing it will make the cells attach even better.
Thanks, Tom. Here's another question coming through in the Q&A section. Can you please just kind of reiterate or explain again how you actually determine cell seeding efficiency when you put them on the scaffolds?
Oh, for sure. Basically, we see the cells on the scaffold, and on the bottom of the plate, we let the cells stay there, and after 8 or 12 hours, we see how many cells remain on the surface. We wash it. We make sure that we take all of them, and we count them and compare it to the number that we see there.
Thanks, Tom. A question here from Alan. Can this be bacteria and fungus free? Like in theory, can you store it at room temperature for food distribution purposes?
Yeah, that's a good question. I believe it is, because if we also made some of these experiments in antibiotic-free environment, we didn't try to do everything antibiotic-free because it's just a waste of time when you do it in the lab. But theoretically, yes, because you don't have bacteria there, and we see the cells, and we see if bacteria didn't go throughout the experiment, it is totally bacteria-free, because bacteria love the media that the cells live inside. So theoretically, you can have a much higher, much longer shelf life because of that. So this is a good point.
Another question here. So in order to replicate tissue more accurately and to provide the tissue with more sponginess or elasticity, what is the deposition of collagen compared to native tissue? Is it around 25%? Do you think that adding collagen fragments to the surface of edible scaffolds could solve the problem?
So first of all, I don't know. I would say that collagen is one of the factors there that is important for the rigidity of the structure, but you need other proteins as well, some kind of more elastic proteins to provide the sponginess, the stretchiness that you require. We didn't try to quantify the amount like in percentage. I think this would be important. And I think that in order to develop something like this in a more wholesome way, again, generating the ECM from several different type of ECM producing cells will have something that, in my opinion, will be more similar to the native muscle tissue. The advantage of these supporting cells is they can grow in any environment. They just grow. Like you can just put them on the floor and they will go. No, I'm kidding. But this fact that basically if you add a different cell, other cell types like the endothelial cells, they are much more spoiled. And because of that, you need to have a specific media specifically designed for them. And fibroblasts and that, no, they don't need it. So you can just have like these 5 different fibroblast types in order to generate this very wholesome ECM that will really resemble what you aim for. But again, generating this very high concentration of ECM, that's something I didn't check in my research, but that's something that is important to check. I agree.
Awesome. Thank you, Tom. That's a research idea for those out there who are curious to explore more deeply.
I will just note we have many more ideas coming up next in our ASAP project, the industry building project. We'll have so many other ideas, and if anyone wants ideas for the research, I'll be happy to share additional ideas. Just let me know, and I will be happy to counsel you on them.
Thanks, Tom. Yeah, Tom is a wonderful advisor for research ideas and very creative too. Here's another question from Varsha. Also, hello, Varsha. Varsha is one of our student leaders for the Alt Protein Project coming out of CU Boulder. And Varsha asks, often to achieve cell attachment on biomaterials, we include ECM proteins or peptide mimics in our scaffold formulations. Do you have any ideas to what the cells are actually attaching to during TSP culture? Are they attaching to their own deposited protein, or are there proteins in the TSP that allow for integrins to attach?
Yeah, so we didn't check this. We just cultured the cells and we saw that they go. So I really don't know if it's the first case or the second. I think in our experiment, we try to see, at first they surely attach also because we seeded the cells on the TSP also with fibrin. However, I can say, so they can obviously attach because of that. However, I will also note that we also seeded the scaffold also without fibrin at all, and we got the exact same results. So maybe that's not that necessary in that stage. But it's a good question to try to think what exactly they are attaching to, whether it's something that they will deposit or not. It's a good question.
Okay, awesome. Here's another question coming from Christoph. First, awesome work. Thank you for sharing. How do you envision scaling cell culture in such scaffolds? Do you think this will require specialized bioreactors?
Yeah, so first of all, you would culture the cells. You will have the cells cultured in a proliferation bioreactor before you even start using this scaffolding. So you will get as many cells as possible. And for scaling this kind of scaffold, I would think of something of a perfusion bioreactor. Perhaps a hollow fiber bioreactor could be relevant for something like that. If you can generate these cylinders of TSP and perhaps punch a hole inside in the middle of them, that could be relevant. One of the interesting facts about the TSP is that it's a sponge-like structure that you can really squeeze. And because of that, that would be a really interesting way to perhaps try and replace the medium that way, because you can just, also when we see the cells, the way that you do it, you put the cells on top, you just squeeze the scaffold and all of the liquids come out and come in, and that way the cells go inside. So theoretically, that might be also relevant for media, removing all of the media and replacing basically the media. But my thought is just a hollow fiber bioreactor or something like that.
Thank you, Tom. A couple questions here about Matrigel. So this first question is, how did you manage to remove Matrigel and other carinogen from your media? Did you add any replacements or cells just survived without them?
We just removed it and it worked not similarly. Like the Matrigel itself is something that is wonderful for the cells. The cells go really well on it, but you cannot eat it. It's some kind of a deposition from mouse cancer cells. No one would want to eat something like that, but it's very good for the cell. We just removed it and we saw that the results are still good enough. The carcinogenic materials were mainly there in order to kill cells that didn't form myotubes, just to increase the myotube, the fusion index. The fusion index is the rate between the number of nuclei inside the fiber and outside. So basically, they just added some kind of material that kill all of the cells that can proliferate. So for us, it doesn't really matter. We don't really want to have something like that. So we just removed it. didn't do anything else besides changing the media composition to make it more optimal for the cells, as we saw.
Yeah, this other question from Theodora, she says, you said that matrigel is not good for cultivated meat. So in your opinion, what is a good alternative for coating?
Yeah, so this is a great question, because I think that basically not just the matrigel, also the fibrin that we used in our work, this is also fibrin is not something that you can use later on. We need to find a good hydrogel, hopefully a protein-based hydrogel, and this is something that would be really nice to develop. As you see, there are basically we need to generate. If you look at the structure, maybe I will share the screen. I closed the presentation. 1 sec.
No worries.
Let me open it again. Yeah. So if you look at the structure of the meat, you can see that the structure, it has these 2-3 layers of the connective tissue: the perimysium, the outer layer, the perimysium, this pink layer over here that has some kind of a honeycomb structure of around these pores, and these pores have the endomysium inside them. So you have the perimysium, this pink area here, the perimysium, and the more reddish pink area here, which is the endomysium. We can say that the TSP itself is something of should be rather similar to the perimysium over here, and we want to culture the cells inside the pores of the scaffold. And we want to initially have some kind of hydrogel that we add inside here to replace the endomysium. So it will be really good. It will be really, usually we use just the fibrin. However, the fibrin is sourced for animals or for humans. And it would be really nice if someone can develop something like that from plant proteins. Theoretically, we can see other hydrogels forming that way from basically, there are, maybe I'm going off topic here, but theoretically, maybe it will still be interesting for people. Basically, this hydrogel should have several, basically it should form gels in conditions which are cell compatible, in low temperature and so on. One of the ideas is there are some kind of solutions which are liquid in low temperatures, and they solidify around 37 degrees. For example, collagen and some kind of ECMs that come from cells. Another example is the matrigel itself. So if someone can generate this gel that is liquid in low temperature, a plant-based protein gel that is liquid in low temperatures, and become a gel at around 30 degrees, 37 degrees, that's something that would be very, very useful for the cultivated meat industry. So that's something, this question just bring me back to these good research ideas that I want someone to do here. So it was just a good excuse to mention this as well.
Yeah, no, that was fantastic. I think we all appreciated the segue. Okay, so here are a couple questions. We have 17 questions left. I'm going to combine 2 of them. A couple questions about kind of whether or not you intend for TSP to remain in the final product. Can you hear me all right, Tom? Oh, you're on mute.
Can you ask it again? I couldn't hear you.
Yes, absolutely. Can you hear me now? Okay. Yeah, so the question is, it's a couple questions about kind of like taste and how TSP affects the taste. So from Katie, is the intention for the final meat product to include the textured soy protein? Is there concern about how that affects taste and texture? And if so, what are next steps to refine this? You're also on mute.
I didn't hear your question at all. I have a very bad connection right now. But I can see the question. Is there an intention for the final meat product to include TSP? Yes. So what we are doing is some kind of a hybrid product. Basically, our logic is that you need a scaffold. you need a scaffold. And if you have a scaffold, at least have a scaffold that is not a plastic scaffold or something like that, but something that will resemble meat already. So if we take a meat substitute and use it as a scaffold, it will be the best scaffold out there in terms of the nutritional value and in terms of the texture. So the TSP itself doesn't have any taste, at least I don't know. I eat it for a long time. That's how I came up with this idea. We just eat it for a long time, and then you realize, wait, this looks exactly like our scaffold. Why not try? Yeah. So ideas from the kitchen, and basically it will be part of the final product. But again, this is something that we probably have to, because you need to start with something. And unless you are using a very long and a very difficult procedure where the entire scaffold degrades in a very regulated manner, that's something that you will have to have at least at these stages.
Thank you, Tom. Another question was whether you've tasted it, and if so, whether you feel like it tastes more like beef or textured soy protein.
So I will tell you that I haven't eaten meat for like 27 years. So I don't know what the taste of meat is. I'm sorry. So it would be a really bad idea to let me try it because it costs so much and I will taste it and you will ask me if it's similar to meat and I would not know. So other people tasted it and they claim that it has the taste of meat, but I'm not a person to ask. I will also note that during the research itself, we still use the FBS inside the product. So at the end, also in the company, there will surely not be FBS, but during the research phase, it still wasn't totally clean or totally cultivated. So I hadn't had the opportunity to try it myself, and I would think it would be more efficient if other people will try it rather than me.
Thanks, Tom. There is a question here from Ning Xiang. Hi, Tom, great presentation. The protein-based scaffolds always have autofluorescence. I would like to know how you deal with this issue to differentiate cells and scaffolds under the fluorescence microscope.
Yeah, so basically doing all of the washes really well, making all of the staining very well, using a high level of laser and a very good confocal resolution. Really inefficient way, but that's how we did the measurement on the TSP scaffold. And also choosing wavelengths that had a low autofluorescence. The main autofluorescent was in the DAPI, and in the red lasers, in the green lasers, we had less autofluorescence. So basically choosing the right lasers and trying to avoid and trying to just do the best we can. Because of that, we just had the high variability in our results.
Thank you, Tom. A question here, how do you actually determine the pore size of your TSP scaffolds?
So basically, maybe I can share the screen again. Basically, we used the two ways in order to look at the scaffold. One of them was with the micro-CT, and the other one was with the confocal microscopy. Basically, we utilize the fact that the scaffold itself has autofluorescence, and we put it on the side where the pores are aligned with the confocal microscope. We tried so many ways to quantify the pores, but at the end we realized we can just put it under the confocal microscope, and if we take those pictures in a very high resolution, you can actually see the pores themselves. So we just visualize lots of samples and quantify that way the pore sizes. As you can see here, those black areas. Here you can see it also, you don't see here one slice, you see the maximal projection, but if you see each slice, it's very clear where there is a pore and where there is not. You can really see the walls of the TSP. inside those images. So basically just using ImageJ and very, very sad students.
Awesome. Thank you, Tom. Here's a question from Stacy. At the end of your co-culture experiments on TSP scaffolds, did you estimate how much of the final construct was TSP as compared to new cell and tissue mass?
Yeah, so we don't really know. We can kind of estimate it using the weight of the scaffold, but I don't really know. I don't really know the answer. I hope that the company will have better answers than me at the future stage.
Thank you, Tom. Another question here. Have you tried growing monoculture of bovine myosatellites along with collagen 1 instead of co-culture and compare the cellular properties?
And compare the what?
The cellular properties.
We haven't, and that's an interesting idea. It's a good question where the collagen will come from, but that's a great idea to maybe start try doing something like that. Perhaps I would say that perhaps not just the collagen. The problem is that ECM doesn't have just one material inside it. It has several different types, and the cells don't just produce collagen. They also do other things. They degrade the ECM that is already in there. They remodel it. They generate different molecules there. For example, some kind of heparan sulfate that can attach to growth factors and regulate their concentration around to the cells around them. So there are many other attributes that the other cells that the supporting cells do besides just generating the collagen. So I would say that this is a nice direction. However, because, as I showed before, because cells have such complex interactions and the ECM has so many molecules inside it, I think that the best way is not to try to generate this ECM by ourselves, but let the cells do it, because they have there are so many processes happening there.
Yeah, thank you, Tom. We have 12 questions left and 14 minutes on the clock. How are you feeling? Do you feel good? All right, sounds good. Here's another question. We'll just go down this list from Varsha. How do the Young's modulus and UTS translate in terms of taste and texture?
So the UTS tells us how rigid the scaffold is. It was very interesting for us because we didn't want to have brittle scaffolds. I don't know how directly it relates to taste and texture. It's just that you have something that is rigid, that doesn't fall off, it doesn't break. The Young module is something that tells you how much the scaffold can be deformed. I think that perhaps there are other mechanical properties, as was shown in the last Cultivated Meat Conference in David Kaplan's lab. They show the other techniques to measure. There is a problem of measuring meat qualities in small samples because no one really did it. And usually these machineries are for samples much higher of size, but specifically for if you talk about chewing, you want to use some kind of shear force. So in David Kaplan's lab, they use something that tries to lower the scale. If someone wants, I will be happy to share the notes. But the Young modules just tells you if you are in the same ranges of deformation that the scaffold can deform in this, whether if you have something that is plastic or something that is more similar to meat or something like that. But I think that there might be other mechanical properties which are more suited to this kind of scaffold.
Wonderful, thank you. There are a few questions about co-culturing that I'll give you in rapid succession. The first is, what are the main roadblocks to co-culturing fat and muscle cells?
Oh, that's a wonderful question. I can say that in our course, we have a course about alternative proteins in the Hebrew University of Israel. And in it, Mark Post came to give, and I told him I want him to talk because in my research, we didn't work with adipocytes at all. And this is very important for this research. Basically, in my research, I try to develop these research ideas that will be synergistic with Marcos to do things that are not repeating what he does, but that can support his results, perhaps because I saw that he's using bovine satellite cells, so because of that we use bovine satellite cells as well. And we saw that he's starting to do some kind of fat experiments. So I said maybe like we can try to go towards the fibroblasts, the ECM producing cells. I would highly recommend, we have the lecture recorded and it is online on YouTube on the GFI Israel channel. I would highly recommend watching it if you want to ask questions about adipocytes and co-cultures. Mainly, I would say that the differences are because the adipocytes have longer maturation process compared to the muscle cells, and probably the media for the differentiation for each cell type is rather different. For the muscle, you want some kind of a media that is a starvation media. I'm not sure what is the right combination of media for the fat, but I guess it's rather different. But I would really recommend talking with Marcos about this. He's extremely nice and extremely collaborative. I'm sure he will be happy to answer. And again, try to watch his lecture. It was really amazing.
Awesome. Thank you so much, Tom. I think that answered most of the questions actually that wanted to know about co-culturing fat and muscle tissue. Do you have future plans to include adipocyte progenitors in the co-culture formulations?
For me, no, I'm not doing research anymore. I'm at the GFI doing all of this counseling and all, but I think this is a really good direction, basically having those for fat, for sure, you need the progenitor cells because the adipose cells cannot proliferate. But if you can take those precursors for the fat cells and the precursor for the muscle, and you can combine them together, maybe that way you can form something like that. Or perhaps even have one cell type, some kind of mesenchymal stem cell that can differentiate into the different cell type. A good idea is maybe to try to generate different areas in the scaffold. Adipocytes, they like smaller pore sizes, so perhaps try to utilize the scaffold geometry and have different areas. One of them will be more suited for the differentiation of fat and the other for the differentiation of muscle. I hope I answered the question and I didn't miss the topic because but yeah, for the co-culture, I would just expect it to have different niches for the different cell types, as they are inside of the skeletal muscle tissue.
Mm-hmm. And Claire also dropped that link, I believe, to Mark Post's lecture there in the chat for all of the attendees to watch and learn more. Awesome. Thank you, Tom. All right, 9 questions left. I think we can do this. A question from an anonymous attendee. In the beginning, there was the depiction of PLGA and fibrin. Is the TSB also supplemented with fibrin?
Yeah, so as I said, in our experiment, we usually use fibrin just because we know it works, and we just didn't want to waste time on trying to check whether we had so many questions to ask, so we left that aside. Although we did have an experiment that compared with fibrin and without it, and the result was exactly the same. So I'm not that sure about the use of the fibrin, but it didn't generate any issues, so we just went on with it because we didn't want to waste time on another small question to answer before we publish our results.
Yeah, that makes a lot of sense. Also, I should say that the chat is blowing up with people who really love your kind of asides. They say, thank you so much for your insightful answers and your very thoughtful answers. So yeah, lots of love for you in the chat. Here's another question from Stacy. Do you have ideas on how you might increase both myotube coverage and alignment on these types of scaffolds?
So I think one of the direction is perhaps to generate a TSP or another protein-based scaffold that has a better geometry. Try to optimize if we talk about the TSP, perhaps optimize, we just use things off the shelf from companies, but if we can optimize the extrusion process, so we'll have pose that are exactly what we need, I think that's something that could be very relevant for us. So we said to increase the myotic coverage and alignment. So this is basically what I think will be best, just to optimize that. Or use another protein scaffold. For example, things like satan could be very relevant. Satan which has much stronger. At the beginning, we had this photo of I went to compare TSP to satan, but at the end we said no, we need to have two types of TSP because we want to check really the TSP. But the satan as well has much really good properties of texture, which are more similar to meat, and it's stronger, so I think it might have less degradation. Just thoughts, but it could be made from other, basically designing the scaffold better for cultivated meat.
Yeah, that's
Another
really great.
for someone.
Yeah, another research idea for those of you out there looking for one. This is also a related question. I think that's the perfect segue into this question. Have you thought of testing protein isolates from other plants as a scaffolding material, such as pea protein? Oh, you're on mute.
Again, the noise, I couldn't hear you. Can you repeat the question? I just moved apartments and now I have these problems with connection issues. Sorry for that.
Oh, no, no worries. We're hearing you perfectly clearly. So Gonzalo asks, have you thought of testing protein isolates from other plants as a scaffolding material, like pea protein, for example?
Yeah, I think that could be a great idea, comparing different types of scaffold, the satan, the TVP, the different types of TVP from different sources, from different plants. All of these seems really great ideas for research proposals.
All right, great, Tom. 5 questions, 5 minutes. From Vin, how do you think cells are surviving in the 1 centimeter thick TSP when technically the nutrients should cross the cell layer?
Yeah, so we didn't try to culture cells for a long period on a 1 centimeter thick scaffold. We just put the cells inside it and just made sure whether the cells are also are seated in the middle or in the outside. If we want to culture cells on thick samples, we need to have a perfusion bioreactor that will perfuse the media through the sample, through those large pores. The medium can flow through there and then diffuse into the cells themselves.
Mm-hmm. Thank you, Tom. Does the presence of the soy protein affect your proteomics sample processing protocol or the data? I would imagine the abundance of soy protein would be much higher than the bovine protein.
This is exactly the reason why we didn't use TSP in those experiments and use the PLLA scaffold. As I mentioned, for the proteomics, we didn't want to use a protein scaffold exactly because of that. And the thought was perhaps to use transcriptomics. However, transcriptomics will not be useful when we try to measure accumulated proteins like the ECM. So basically, we went on doing the proteomics on the PLLA scaffold just because it was simpler and easier to get results. Because we were really interested in the how cells affect one another and not exactly to get the results related to the TSP in that sense.
Mm-hmm. Okay. Did you use the 2 types of TSPs as is, or did you do some kind of modification before the deposition of cells?
Nothing, just use it as is. We use these TSP flakes. TSP can come in very different geometries. So we use TSP flakes, which are about 2 millimeter in size, and we just cut them into the 6 millimeter cylinder. So it will be kind of repeatable, and we try to use TSPs which have the same height.
A question from Aaron Reese Clayton, our beloved colleague. Based on these experiments with bovine cells, do you think similar results might be observed with growing other species of cells on TSP scaffolds, such as chicken or fish?
So the reason that we use the bovine cells is because 99.999% of, well, I wouldn't go, 99% of tissue engineering is done with mammalian cells, with humans or with mice. So we thought that going for bovine will be better because we know what is really going on there. For other species, I think it would be possible. However, the farther the species is, the more research we'll need to do in order to understand better how tissue engineering will work with those species. Because of that, I urge everyone to try to work with, because cultivated meat is it's a startup idea. It can happen, and it can also not happen. And because of that, it's really important that we'll have as much research as possible so that the first products will be good. And because of that, I really think it's important that Mark and others try to use bovine cells, because it will just be simpler in my mind. And but I think that after we can get the first products out there, we can translate the information to other species, and I think it can theoretically work, but we will see. Hopefully, we will see it soon in other species as well.
Thank you, Tom. The final question, and then I'll let you close with some final thoughts, from John Yuen: was it ultimately worth it to include endothelial cells, or do you think the smooth muscle cells were really sufficient?
Yeah, so I don't think the endothelial cells did much. They are expensive. They need specialized formulations, and at the end, we didn't see much that they had a very good value for, at least in my mind, they didn't have very good value. I would recommend to focus on the supporting cells, the muscle cells, and on the fat cells. Those are the key components. We hope that the endothelial cells would mature the tissue faster, but we didn't see much evidence for that. There was a little bit evidence, but in my mind, because we want to make it as simple as possible, as cheap as possible, adding the endothelial cells really, I don't think that the value of them was as good as it should have been to make this worth the effort that they require.
Thank you so much, Tom. We've reached the end of our question queue. This has been such a phenomenal presentation. People are saying in the chat it was super interesting and thoughtful, and someone said gorgeous talk. Thank you so much. Tom, do you have any final thoughts that you want to leave off this community with?
Sure. So first of all, thank you all for being here. It's amazing to see how many people care about this type of research and are interested in it. So go and do this kind of research, because it's just phenomenal how much we can change the world just by developing all of these amazing new techniques, both in cultivated, plant-based, and in fermentation. And basically, we see that this industry is really booming, also especially in Israel. It's really nice to see how we can really change the world and make things better that way. If you need any kind of counseling regarding cultivated meat, I will be happy to support anyone. You are welcome to send me an email, and I'll be happy to try and support you at least as much as possible. And good luck for everyone with the companies that you are forming and with the project that you are forming. We really wish you the very best of luck, and hope that one day all of you or some of you will come here and give lectures as well, because why not?
Yeah. Thank you so much, Tom. Yeah, that's a great way to end off. Everyone, I've dropped Tom's email there in the chat for you to access if you have additional questions or research ideas that you'd like some counseling on. Thank you so much for joining us today. It's always so wonderful to see you all come out from every corner of the world. To answer some of the questions coming through about whether there will be a recording, yes, there will be a recording of this seminar posted on our YouTube channel. It will be added to this playlist with the rest of our seminars as part of our scientific series that I've just posted there in the chat as well. So we look forward to seeing you all again next month for Dr. Amy Rowett from UCLA. It's a perfect follow-up, actually, to Tom's seminar. She'll be talking about using hydrogel scaffolds to create a marbled kind of texture in cultivated meat. So we're looking forward to seeing you all there. This was fantastic. Thanks again, Tom, and we'll see you all soon. Bye.